This video will cover the cell cycle, including its stages, mitosis and its phases, meiosis and its phases, the differences between meiosis 1 and 2, the purpose of meiosis, and the differences between mitosis and meiosis. The cell cycle is a series of events where a cell grows and divides, replicating to make two new cells. This process replaces damaged, dead, and worn-out cells. Most body cells divide by mitosis, resulting in two genetically identical daughter cells. Gametes, however, are formed by meiosis. The cell cycle includes two phases, interphase and mitosis, or M phase. Interphase is where a cell spends most of its time. During this phase, it grows, replicates its chromosomes, and prepares for cell division. After interphase, the cell undergoes mitosis. The resulting daughter cells then enter their own interphase. Interphase has three stages. The first gap phase, G1, synthesis of DNA S phase, and second gap phase, G2. The first gap phase, or G1, is when the cell grows in size and volume. This is often the longest and most variable phase in length. Sometimes, cells may enter a resting phase, called G0, instead of continuing the cell cycle. During the synthesis of DNA, or S phase, chromosomes replicate, forming two identical copies of DNA. After the S phase, the cell has enough DNA for two cells, nearly ready for division by mitosis. The second gap phase, or G2, occurs after the S phase. In G2, there is further growth and preparation for cell division. After the G2 phase, the cell is ready to divide, and mitosis begins. Mitosis is a continuous process with four distinct stages: prophase, metaphase, anaphase, and telophase, which can be seen by light microscopy. During prophase, the replicated chromatin coils tightly and becomes easier to see. Each chromosome is paired with its copy, forming a double chromosome unit joined at the centromere. The mitotic apparatus appears, and the nuclear envelope disappears. In metaphase, the chromatids align on the center of the spindle, attached by their centromeres. During anaphase, the centromeres separate. One of each pair of sister chromatids migrates to each end of the spindle as the microtubules contract. In telophase, the mitotic spindle disappears, the chromosomes uncoil, and the nuclear envelope reforms. Following telophase, cytokinesis occurs. This involves the splitting of the cytoplasm, intracellular organelles, and plasma membrane, resulting in two identical daughter cells. Organelles develop further during interphase. Now let's discuss meiosis. Meiosis is a cell division process where a single parent cell divides twice to produce four independent daughter cells, each with half the chromosomes of the original cell. Meiosis produces gametes. Unlike mitosis, meiosis involves two distinct divisions and results in four non-identical daughter cells, which is the basis of genetic diversity. Meiosis involves two stages, meiosis 1 and meiosis 2. Each includes nuclear division, or karyokinesis, and cytoplasmic division, or cytokinesis. Before meiosis 1, cells undergo a growth period called interphase. Interphase for meiosis includes the G1 phase, where the cell increases in size and synthesizes RNA and proteins. The nuclear envelope remains intact, and chromosomes are chromatin fibers. The S phase is the period of DNA synthesis, where genetic material is copied. Each chromosome duplicates into two sister chromatids. The G2 phase follows, where the cell continues to increase in size with further synthesis of RNA and proteins. The first meiotic division, meiosis 1, produces two genetically different daughter cells. DNA replication occurred beforehand. Chromosomes exchange genes in a process called crossing over. Following crossing over, chromosomes separate, with a random assortment of maternal and paternal chromosomes going to each daughter cell. This process explains the inherited combination of characteristics. Chromosome pairs separate, guided by a spindle, and the cytoplasm divides, resulting in two genetically unique diploid daughter cells. Prophase 1 is the longest phase. Duplicated chromosomes condense, forming a tetrad with homologous pairs. Crossing over occurs, where homologous chromosomes exchange DNA at chiasmata. Spindle fibers attach to the centromeres, the nuclear envelope breaks down, and chromosomes move towards the center. In metaphase 1, homologous chromosomes align along the center of the cell. Centrioles are at opposite poles, with spindle fibers extending from them. The centromeres orient toward the opposite poles. During anaphase 1, homologous chromosomes with two sister chromatids separate and migrate to opposite poles due to the contraction of spindle fibers. In telophase 1, chromosomes stop migrating, resulting in a haploid number at each pole. The nuclear envelope forms. The spindle fibers disappear, chromosomes uncoil, and the nucleolus reappears. Cytokinesis Y involves the division of the cytoplasm, forming two individual daughter cells. In most cells, this occurs simultaneously with telophase 1. The second meiotic division, meiosis II, follows. The two daughter cells are formed with half the number of chromosomes. This division further halves the genetic material to produce gametes. The centromeres separate, and sister chromatids move to opposite ends, resulting in 4 haploid daughter cells. In prophase II, the nuclear membrane breaks down, and spindle fibers reappear. Centrioles divide, forming 2 pairs. Chromosomes migrate towards the center without further replication. During metaphase II, chromosomes arrange on the cell's equator with the help of spindle fibers. Centrioles are at opposite poles. The centromere divides, creating daughter chromosomes and spindle fibers attached to each. In anaphase II, the daughter chromosomes are pulled toward the opposite poles by the spindle fibers. At the end of this phase, each end of the cell has a complete set of chromosomes. Telophase II involves the formation of the nuclear membrane around each chromosome and the disappearance of spindle fibers. The nucleolus reappears as the cell prepares for cytoplasmic division. Cytokinesis II is the second cytoplasm division, forming 2 individual daughter cells. This process is identical to cytokinesis I. The result is 4 non-identical haploid daughter cells, each with half the original chromosome number. In meiosis I, homologous chromosomes separate, resulting in 2 diploid daughter cells. In meiosis II, sister chromatids separate, resulting in 4 haploid daughter cells. Unlike meiosis I, no genetic recombination or crossing over occurs in meiosis II. The purposes of meiosis include maintaining the correct chromosome number in organisms by forming haploid gametes. Meiosis also creates genetic diversity through the exchange of genetic information, which is key to evolution. Additionally, it helps repair genetic defects through recombination. Next, we will review the differences between mitosis and meiosis. Thank you for watching this video on the cell cycle and its divisions.